Hi guys, my name is Ian, and in this video I'll be going over the structure of monosaccharide glucose both in alpha and beta form, as well as the structure of maltose, which is a disaccharide formed when two alpha glucose bonded together. So, in glucose there are a total of 6 carbon atoms, colored in gray in the model here. In addition to the 6 carbon atom, there are also 6 oxygen atom colored in red and 12 hydrogen atom colored in white. In this model, glucose is presented in a ring structure, which is why you can see 5 carbon atoms and an oxygen atom forming a ring. This is the oxygen atom that I had just mentioned. Meanwhile, this is the first carbon atom, this is the second, this is the third, this is the fourth, and this is the fifth. Notice that I had only mentioned 5 carbon instead of 6. This is because the 6th carbon atom is here bonded to the 5th carbon atom, as well as with 2 hydrogen atoms and a hydroxyl group. Otherwise, each carbon atom from carbon 1 to 4 are each bonded to a hydrogen atom as well as a hydroxyl group, with the 5th carbon being the exception, bonded only to the 6th carbon atom and a hydrogen atom. Like mentioned before, glucose exists in 2 different types, alpha and beta. They are isomers. The difference between the two is located at the position of their hydroxyl group bonded to the first carbon atom. In alpha glucose, the hydroxyl group is below the plane as the first model displayed. Meanwhile, in beta glucose, the hydroxyl group is above the plane, as the second model displayed. When 2 monosaccharide, which in this case 2 alpha glucose bonds together, they form a disaccharide called maltose. As you can see from the third model here, this is the first glucose and this is the second glucose. The first glucose and the second glucose are bonded together through this oxygen atom in the middle. What happened here was a process called dehydration synthesis. If you look thoroughly, you will notice that the bonds to carbon 1 on the left model and the bonds to carbon 4 on the right model are different from their original. Together, they are missing their hydroxyl group. In dehydration synthesis, a water molecule is formed from a hydrogen atom from one of the monosaccharide and a hydroxyl group from the other, creating a H2O molecule that breaks apart from the glucose. As a result, the 2 glucose creates a new bond between themselves called glycosidic linkage through a covalent bond from the leftover oxygen atom of one of the hydroxyl group. And due to the linking position of carbon 1 and 4, the glycosidic linkage is also termed as the 1,4 glycosidic bond. Dehydration synthesis is a process that is reversible. 2 alpha glucose monosaccharide can be reverted back to their original structure through a process called hydrolysis. In hydrolysis, a water molecule is added back to the glucose, breaking down the 1,4 glycosidic bond and reverting them back to their original structure. And as a matter of fact, this process is normally carried out by enzymes in cells.